Hi, my name is Maryam, and I'm excited to present my research about accumulation and metabolism of endocrine disrupting chemicals in a rat model and its impacts on brain and tissue distribution. Our study classes of endocrine disrupting chemicals, including microplastics and PFAS, on tissue distribution and potential impacts on neurodevelopment. To give you some background information, I should say endocrine disrupting chemicals such as microplastics and PFAS are persistent pollutants that can cross biological barriers like the blood brain barrier and accumulate in various tissues, including the brain. Microplastics, particularly polystyrene particles, have been shown to bypass the intestinal barrier and blood brain barrier within hours of exposure, potentially contributing to neurodegenerative changes. Similarly, PFAS are known to accumulate in specific brain regions such as the brainstem and hypothalamus, raising concerns about their impacts on neurodevelopment and endocrine function. For our study design, we used 30 rats with a 1-to-1 male to female ratio. The rats were divided into control and case groups with a 1-to-4 ratio. PFOS was injected at three dose levels of 0.1x, 1x, and 10x at 20 microgram per kilogram. Samples of whole blood, liver, and cerebellum were collected after killing the rats at 89 to 96 days of age, and the samples were stored at minus 40 Celsius until the analysis. For the analytical methods to analyze microplastics, we used digestion with potassium hydroxide, followed by microfiltration through a glass filtration unit, and pyrolysis GCMS was employed for detection with precise temperature settings to identify polymer types. For PFAS, we used homogenization and solid phase extraction followed by LCMS-MS analysis using MRM transitions from EPA method 1633. These methods allowed us to quantify the presence of microplastics and PFAS in biological matrices with high accuracy. In terms of our results showed that PFAS concentrations ranged from 0.45 to 141 nanogram per milligram and with an average of 21 nanogram per milligram. PFAS levels were highest in the liver across all groups due to their strong affinity for protein binding, and male rats exhibited 1.8 to 2.6 times higher PFAS concentrations in the liver compared to female. The bioaccumulation factor, however, decreased significantly at higher doses due to saturation of binding proteins and elimination dynamics. For microplastics, concentration ranged from 57 to 700 ppb with an average of 350 ppb. The highest microplastic concentrations were found in blood, with polycarbonate being the dominant polymer type. Interestingly, female rats exhibited higher microplastic concentrations than male in most cases except for polystyrene. However, no significant correlation was observed between total microplastics and PFAS concentrations in blood samples. So, to discuss more our results, I should say that these findings highlight distinct accumulation patterns for microplastics and PFAS in biological systems. While PFAS showed a strong tendency to accumulate in the liver due to protein binding, microplastics were more prevalent in blood. Additionally, our results suggest that microplastics and PFAS may follow independent pathways of bioaccumulation despite their co-occurrence in tissues. And for future studies, we aim to explore the synergistic effects of microplastics and PFAS on endocrine function by studying their combined impacts on phenotypes. So, as acknowledgement, special thanks go to our collaborators from UT Austin and my colleagues at UTA for their invaluable contribution. Thank you for attention, and I would be happy to answer any questions you may have about this research.